And just so you guys know, I won't be doing like a live Zoom for class. So if, yeah, but if you end up, on quarantine or isolation or whatever throughout the semester, then I can always send you the recording of the class for you to watch. But yeah, there's a few reasons I don't want to do the live Zoom. One of them is it's just really hard to manage people in two places. The other is I don't want people. I noticed in the past when I've done that, people start seeing like, oh, there's people on Zoom. Maybe I should just stay home today too, and I'm not feeling too good or whatever, So it kind of just makes it a little bit easier to do that. So I just do the recording instead of the live Zoom. But you can still get all the information you need that way. So yeah. All right. Okay, so we kind of left off last class talking about how we could use the same muscle to get a different amount of force out of it, right? How we vary the amount of force we can get in a muscle. We talked about this idea of we can use a differing number of motor units to do that, right? So we can activate more or less motor units, and that happens via activating more or less motor neurons, right? But then we also talked about how within each motor neuron, we can change the frequency of the action potential, right? If we get a higher frequency action potential, then we get more, end up getting more acetylcholine release, which leads to more calcium release, which leads to more active myosin binding. So within that single muscle cell, it gets more force, right? And so this slide will kind of walk us through that a little bit. So if we think about the idea here where we kind of have this group of neurons, maybe somewhere in our prefrontal cortex, where we actually like think about contracting muscle, right? There's some kind of conscious thought like, all right, I'm going to move myself this direction. And again, so yeah, that would be kind of be represented by this, okay? And then we have another group of neurons that would be in our primary motor cortex. And the way it typically works is we'll have like one part of that part of our brain that's dedicated to the quads, right? And another part of that part of the brain is dedicated to biceps, right? And so there's kind of this little group of motor neurons that are all for the same muscle or muscle group, right? And so the idea would be that we're getting some kind of a stimulus that's going to start in somewhere where we think about contracting the muscle, that's going to move back to the motor cortex, and then it's going to activate a certain number of motor neurons there for us to contract, right? And so if we think about and so those would obviously all be interneurons, right? Going from neuron to neuron. And then the motor neurons there. So we'll go down to the muscle cell. Okay. So if we start out kind of thinking like a low strength stimulus, right? Something we know we need to just move, we're just going to take maybe we'll say we'll just take a step. Okay. We know we don't need a ton of force to be produced there with that step. And let's just pick a muscle like our quads. Okay. We know we're going to need to kick our legs out a little bit. We're going to need to push off a little bit. So we need a little bit of force out of those quads, not a ton. So we're gonna kind of activate a certain number of neurons here that are going to send their signal back to the motor cortex, right? Primary motor cortex. That's going to activate a certain number of motor neurons, and then those are going to go down and activate the muscle. Okay. And so it's a low strength stimulus. So again, that size principle also tells us that we're going to activate the smaller motor units first, right? So we get the smaller ones. We don't get quite so many, and on each of those we have a low action potential frequency. Okay, so we don't get quite as much acetylcholine release, don't get quite as much calcium release, don't get quite as much active myosin binding. Okay, then if we want to use that same muscle to produce a lot of force, right, then we're going to actually have a higher strength stimulus. So we're actually going to send input from more neurons in the conscious part of our brain back to the primary motor cortex, which then that's going to activate a larger group of motor neurons, right? So we get more motor neurons, which means more motor units, okay? And then on each one of those, first of all, we get the smaller motor units and we get the larger motor units together contracting, right? On each of those, we have a high frequency action potential, right? So then we get a lot of acetylcholine release, a lot of calcium release, a lot of active myosin binding. So we get a lot of force generated with each one. And that's kind of the basics of that. Okay, so any questions about that? And it's important just to remember that, yeah, we've got the two kind of levels, how many motor units are we activating, and then within each motor unit, how high the frequency of action potentials are we targeting. Okay, and we won't necessarily go through this right now, but this could be something you kind of think about and kind of fill out on your own. What type of stimulus are we getting with the lower lower force production needed, small typing? All right, you're using your text a little bit to do that versus actually just like throwing a punch, trying to use your text to hit something as hard as you can. And you can kind of walk through that to review that, but we won't do that right now. We need to move on to the metabolism stuff. But any questions on that before we move on? All right, so let's go ahead and hop over to the metabolism notes. Okay, and so I put this verse in here for this one because I do when I think about this process of creating ATP, which is really what we're going to talk about in terms of metabolism. It is one of these processes that kind of, for one thing, connects us to the rest of the biology of the world, which is every organism in one way or another has to go through some kind of process to make ATP, because ATP is really the currency of energy across all of life, everything from plants to animals to to bacteria, single cell organisms, all the way up to humans. We all have to make ATP to do all of the chemical functions that we do. Now plants do it in a different way than we do, but it is kind of this just kind of incredible process that is really mind-blowing when you get into how all the little steps work. And so to me, it always feels a little bit like holy ground to be talking about this stuff, which a lot of this stuff does in this class, but this one in particular kind of feels that way. So yeah. Okay, one of my favorite parts of this whole thing is this enzyme right here, and that's why I have it on the back of this slide, or the background of the slide. This enzyme, anybody know what that one is? Say it again. ATP. Yeah, ATP synthase. There it is. Yeah, so this is I don't forget to it today, but maybe tomorrow or on Friday we'll watch a little video kind of on this. But yeah, it's just this incredible enzyme that's designed in this perfect way to pretty much act like a rotary engine, which is similar to engines you would see in some of the Mazdas were kind of the earliest cars to use rotary engines. But it has all these parts in it that pretty much spins around and creates 99% of all of our ATP. Is this one single enzyme right here, and its design is really perfect to do that. So we'll get into a little bit more how that works on Friday. So yeah, so just kind of the broad overview of this idea of metabolism is obviously we're taking our three main macronutrients, right? Carbohydrates, proteins, and fat, and we're using those to make ATP, right? And we'll spend most of our time on the carbohydrates one with glucose and glycogen. We won't get too into much detail on how we do this with fats and proteins, but just a little bit. If you're taking sport nutrition, you'll get into that one a little bit more. The other two, especially fats one, a little bit the amino acids. We'll spend most of our time here in this pathway for this class. Okay, one of the things that we'll spend a lot of time talking about is mitochondria, because most of these processes, especially the oxidative system, occur in the mitochondria. A couple of things that I just like to point out with the mitochondria. Number one, we've got all of these little folds on the inside, right? So it's kind of a super folded thing called cristae, right? And what's kind of the one word easy answer as to why there's such a folded membrane inside of the mitochondria? How do those folds help us? It's the same thing when we have our digestive tract, where we have those little microvilli and villi, lots of very, very bumpy thing inside of our digestive tract. Right? We have lots of, we need a lot of this same thing I'm looking for inside of our lungs, which is surface area, right? We need lots of surface area. And the whole reason we need surface area here is so we can plug in a bunch of enzymes onto that membrane, right? The more spots we have that are folded like that, then the more little membrane space we have to plug in ATP synthase, as well as all the other enzymes that run the electron transport chain, Krebs cycle, and all of that. They're all plugged into those to that membrane. Okay. There's something kind of super interesting about mitochondria when it comes to kind of the theories on the origin of multicellular organisms, which pretty much mitochondria look a lot like a little bacteria, right? And so the theory is that, you know, certain bacteria are like us, and they use oxygen to make ATP, and then they produce CO2. But then there's bacteria that do the opposite of that too, right? They take in CO2 and produce oxygen, right? Similar to plants. And so the theory is that, you know, early on it was all single cellular organisms, and at one point a bacteria engulfed another one, and then pretty much just made that other one its slave to make all its energy for it, right? And instead of destroying it, it stayed alive in there and produced the first multicellular organism, right? And then also started producing eukaryotic cells, which means cells that have a bunch of little organelles inside it. Right? So bacteria don't. It's a little bag with everything happens across their own membrane. But eukaryotic cells have all kinds of organelles inside of it. Right? And so this idea of mitochondria kind of being the necessary thing for the cell to be able to produce its own power within itself, kind of one of the earliest things that need to happen to produce a complex cell. So some interesting part of the evidence for that too is that mitochondria have their own DNA, right? And mitochondrial DNA. Certain diseases are connected to mitochondrial DNA only. And we get all of our mitochondrial DNA from our moms, not from our dads, because the mitochondria in the sperm is in the tail and it's lost when conception happens. And so all the mitochondrial DNA comes from mom. But many proteins that can't be encoded by your DNA inside of the nucleus, that DNA is found in the mitochondria. Yeah, so just some interesting stuff. Last thing interesting about mitochondria is this is how we typically see them in textbooks, kind of shaped like these little beans, but they really don't look like that. They don't really look like that at all. What they look like is this kind of big sort of splash of gel, pretty much. And then the more that they grow, we'll talk about how they grow with training and aerobic training, like that, they just kind of expand. Right? Normally we might think of it like sprouting another bean, have all these little beans, but really it's not that. It's just this big thing called the mitochondrial matrix, which is just a group of this kind of stuff, and that's what that little image of the bottom is. So, yeah, some interesting things about mitochondria. One of my favorite books of all time, if you're interested in this stuff at all, is called Mitochondria and the Meaning of Life, Power, Sex, and Suicide. It's quite an interesting title, but it's by a science author named Nick Lane, who writes a lot of cool science stuff. He's a biochemist, but super fascinated about kind of history and all the science around mitochondria. So, yeah. All right. So all of that and all of metabolism is really connected to this idea of ATP, right? So when we think about organic molecules, meaning the organic molecules inside our body, ones that contain carbon, we've got proteins, we've got fats, carbohydrates. What's the fourth one? The biggest one. It's included in here. Also DNA and RNA. What's that category called? Nucleic acids. Yeah, there we go. So ATP is one of our nucleic acids, right? Which one does it look more like here, RNA or DNA? Remember how to tell? What's the R or the D stand for? It's a sugar on the inside, right? There's a sugar group. So DNA has deoxyribose, RNA has ribose. So is ATP a version of RNA or DNA? RNA, absolutely. Yeah. So this is the version of RNA. Okay, and this is the A base that we would see in DNA or RNA, right? Adenine. Right, the A is going to match up with T. Obviously, we're not matching anything up with ATP, but that's the same base that we would see in DNA. Right? So we got that, but then the big difference here is instead of just having one phosphate group like we have typically on DNA and RNA, we've got three. Right, so we've got those three phosphate groups, adenosine triphosphate. Okay. So one interesting fact about ATP, at any instance, each cell contains 1 billion ATP molecules. Right? We've got about 30 trillion cells, so you can count up how many ATP there is in the body at a given moment. But this is only enough to last us about 3 minutes of rest, not an exercise. And so that just means we're constantly producing tons and tons of ATP. couple of us in this room got to go to a conference last semester where we sat in a lecture where we walked through, a scientist walked us through the ATP usage in the 2-hour marathon. Where did he land at, guys? 56 kilograms. Yeah. So the amount of ATP that it takes to run a 2-hour marathon, it can be calculated somewhat simply. And then the molecular weight of ATP and kind of put all that together. 56 kilograms worth of ATP is burned in 2 hours of running at that pace. And so that's just an insane amount, right? I mean, the guy who can, the only man in the world who's ran anywhere close to a 2-hour marathon is Kipchoge from Kenya. And he's probably not much more than 56 kilograms, right? He's probably only about 60 kilograms. So almost his body weight in ATP in 2 hours to be able to run at that speed. So yeah, pretty fascinating stuff. Okay, any questions about that? Okay, so what we do with ATP, when we want to get the energy out of it, go through a process called ATP hydrolysis, right, which really just means we combine it with water, okay? And we have all different kinds of ATPases throughout the body that can do that. The one that we'll talk a lot about, we've already been talking about, is myosin ATPase, right? And so all we're doing is we're taking that ATP, breaking one of those, the last phosphate off, turn it into ADP, so then there's 2 now instead of 3, and we got that phosphate. And those last phosphate bonds are kind of considered high energy bonds, meaning that it's likely for that phosphate group to bind to something else. Right? And it does that, it changes that protein typically and kind of exerts some energy on it. Right? And this is really the way all chemical reactions in our body are driven if they require energy from a primary source like this. So that's really what we're talking about, is how we replenish those ATP stores. That's what we're talking about with metabolism. Okay, so let's talk about the 3 main sources of where we get our ATP. Right, so carbohydrates. How much do we store in the body? Not a whole lot, right? Maybe somewhere around 2,500 calories worth of carbohydrates. Okay, which is something. Where's the primary place we find it? Say it again. Muscle glycogen. Okay, muscle glycogen. Absolutely. That's number 1. Okay, and you can see that here. We have about 2,000 of those 2,500 calories in our muscle. Okay, and then the other one is the liver. Right, the liver is the second largest storage site of glycogen. Okay, and then obviously we have some in our blood. That's just our blood sugar. Right, so it's a little bit there. 60 or so calories worth of glucose in the blood at any time. Okay, fat, we can store a ton of it. All right, so for somebody who's about 70 kilograms and 12% body fat, which is pretty lean, they still have around 70,000 calories of fat in their body. Right, so this is our primary way to store energy in our body, no question. Most of this is in our subcutaneous adipose tissue. Right, that's the fat that we can feel, it's right underneath our skin. Right, and then we've also got visceral fat around our organs. We've got fat. We do have some fat storage in our muscle cells too. And actually, the more somebody becomes endurance trained, the more they will store some fat inside their muscle cells. But then we can also have bad fat storage in our muscle cells, which happens with type 2 diabetes. So kind of which type of fat is stored in there. We can store fat for fuel directly in our muscle cells as well as these other locations. And then protein, how much protein do we store for energy purposes? We got about 70,000 calories of fat, 2,500 in carbs. How much protein? 0, right? There's no place in our body where we just store protein just to store it, right? Everywhere we have protein in our body, it's doing something, and it's almost functional. Okay, doesn't mean we can't tap into that and use it, but it's never just there for storage. Okay, so when we do a long bout of exercise, exceeding an hour, then we're going to see maybe about 5 to 10% of our total energy usage coming from. But anytime we're doing that, we're using protein that was doing something else. Right? It's not like it was just there waiting to be used. It had a function before, so we need to replenish that. Okay. All right. So any questions on that? Okay, so let's talk about the three energy systems here. So we'll spend some time talking about the first one, this ATP creatine phosphate system. Okay, and then we'll spend a little bit of time talking about the glycolytic system, and then finally the oxidative system. Okay, when it comes to glycogen, we kind of already just talked about this. Glycogen, we find it either in our muscle or in our liver. And then glucose, we're just pulling directly from the blood if we're using it for fuel. Okay, and that could have started as glycogen in the liver, and then the liver breaks it down, turns it into glucose, puts it into the blood, and the muscle cells can pick it up. Yeah. Okay, so let's just a couple of questions here. I want you guys to turn to your team, people on your team, and just kind of walk through these two questions. Okay, so just ranking the energy systems first from fastest to slowest. Secondly, from highest level of endurance to lowest. Okay, talk about that for a second. And so the way we'll answer all the kind of group questions like this is just kind of designate one person on your team to be the person who's going to vote. And then I'll just have you hold up a finger. Okay, so like 1 for A, 2 for B, 3 for C, 4 for D. Okay, so first question here. You guys ready? Got your voter? Designated voter. Is that kind? Is she, yeah. I didn't know she was on. Okay. Oh, okay, cool. Yeah. All right. I'm also recording for this too. Yeah. Yeah. So however she wants to do it. Yeah. Cool. All right. Go ahead. Hold them up. Okay. Cool. Yeah. So everybody's saying 3. So definitely the fastest is our creatine phosphate. Second, glycolytic. Third, oxidative for sure. Okay. And then in terms of endurance, go ahead and hold up your answers for that. Yeah, so just the opposite, right? Push your oxidative, so post endurance, glycolytic, so then on creatine phosphate is least amount of endurance. Okay. All right. Any questions about those two ideas? Okay, and this graph just kind of shows that in a way where we see the peak power output of the creatine phosphate system is very high. And we get to that peak super quick within 10 seconds, but then it starts petering out pretty quickly. Okay, the glycolytic system can peak somewhere around 30 seconds, but then right once we get to around 2 minutes, it's already starting to decline. Okay, but then the aerobic system can really kind of get going pretty quickly within 10 seconds or so, but it can go for a long time after that, obviously much more than 5 minutes, which is on this graph, hours and hours. The things that stop these systems from working are all different, right? So the creatine phosphate system stops because it gets depleted. The glycolytic system doesn't stop because it gets depleted. It stops because it has kind of this negative feedback mechanism built within it with the lactic acid. And then the aerobic system will really only stop if we run out of substrate, which really doesn't happen. As you saw, we got 10,000 calories of fat to burn, so that's not really going to happen. So it's usually due to other things that we stop, not because our aerobic system is taxed, right? So, yeah. Okay. Any questions on this? Okay, so let's talk about the creatine phosphate system first. So this is the simplest, just a single reaction, really. So creatine phosphates is a protein, right? When the creatine part's a protein, the phosphate is just attached to it. Okay, so this is really just giving us one ATP at a time. Okay, This one really peaks somewhere around 3 to 15 seconds. Okay. And how many of you are in the lab this semester? Yeah, okay, so good, half of the class. One of the tests you guys will do coming up, and I'm not sure exactly how we'll do it, how it's going to happen with the COVID stuff, and everybody's going to do it this semester. A couple of you are, but somebody will do this test. Wingate test is one that really kind of gets at both the creatine phosphate system and the glycolytic system. And one of the things that if you get to do the test, you'll notice is that so you put the bike at a very high resistance, and you just pedal as fast as you can for that for 40 seconds, or typically the 40-second one in the lab. You can also be done in 30 seconds. But the thing you'll notice is after right around 10 to 15 seconds, you kind of hit this wall. You're feeling pretty good for about 10 to 15 seconds, and your RPMs are usually 150, 160. And then right around 10 to 15 seconds, all of a sudden your RPMs drop to maybe 80 or 90. You just feel like, how in the world am I going to keep pedaling? there's no way I can keep going. And if the people are doing the test wrong and they say how much time you got left, that's not good, because you think there's no way I can make another 20 seconds. but that's because you just lost your creatine phosphate system. That's what that is, right? You just completely taxed out your creatine phosphate system. So now you're fully relying on the glycolytic system, maybe just a little bit of aerobic at that point. But you feel that when the creatine phosphate system gets depleted, you feel it because you're relying on one less energy. But yeah, so this one is very simple. We have an enzyme called creatine kinase, which is the major enzyme involved in this. And what that's going to do for us is get that phosphate off of the creatine so it can bind to the ATP that's floating around. And then we can make ATP. Okay, so that's it. It's just really that single reaction to do that. And so obviously there's no oxygen used in this one. And when you think about this system, what would be some activities that you would really think would kind of exemplify the use of this system? What are some ideas?
Like the strongman competitions.
Okay, give me an example of like one thing they do.
Lift the car.
Lift the car, perfect. Yeah, definitely. Yeah, you need a lot of energy in a super brief period of time, right? Just a matter of seconds. So absolutely. So any kind of power type of lift, power type movement, even swinging a bat, lifting a weight, those kind of things for sure. We need a powerful creatine phosphate system to do that. What else? Outside of just like a single muscle movement. Sprint, yeah, somewhat short of sprint, right? Yeah, 100 meter dash, 50 meter dash, something like that would be definitely a very powerful creatine phosphate system to be fast at those. Okay, for sure. Yeah, and the thing that stimulates this whole system, because in your muscle cells you've got a bunch of this PCR, right? The creatine or the phosphate bound to it. You've got that ready to go. Thing that gets it to start going through this process is the presence of broken down ATP. Right? So you've got a bunch of stored ATP in your muscle cells. You've got a bunch of stored creatine phosphate in your muscle cells. As soon as you start exercising, that stored ATP starts getting broken down into ATP and phosphate. Right? So as soon as we start having a lot of this show up, then that stimulates creatine kinase to start doing its job of just pulling that phosphate off the creatine, making more ATP for us. Right? We'll find that all of the energy systems are really stimulated by things like that, byproducts of breaking down ATP. So okay. All right, so let's talk about glycolysis a little bit. Who can give me the definition of what is glycolysis? Goes from what to what? Does anybody have biochemistry? Yeah. Glucose pyruvate, there it is. Okay, so glucose to pyruvate. Okay. And then it's a part of both what we call the glycolytic system and the oxidative system. And when we're breaking down glycogen, it goes through a process called glycogenolysis. Right? So lysis always means to like break apart, to break down. And so glycogen is just breaking that down, so to break down. And it's going to share most of the same steps as glucose to pyruvate, but it just has a little bit different step early on to to get that process started. But glycogenolysis and glycolysis share almost all the same steps, except just the first couple on the top. Okay, so again, this is kind of a shared pathway between both the glycolytic system and the oxidative system. Okay. All right, so this is just a broad overview of it, right? So we're seeing that glucose to pyruvate. Okay, and then we'll talk more about this, but when it comes to pyruvate, it kind of has a fork in the road after it's formed, right? It can either go through this enzyme, which is called lactate dehydrogenase. Okay, and then it just turns into lactate. And that's the end of the glycolytic system if it goes that route. Right? We're done with the glycolytic system if it goes that way. If it goes to this one instead, the PDH, which is still dehydrogenase, but it's just pyruvate dehydrogenase instead, then it turns into acetyl-CoA, and then we start the Krebs cycle, and then this one takes this electron transport chain, and that's the oxidative system, right? So this is the oxidative side. Okay, and we'll spend a little bit of time talking about kind of what determines the fate of pyruvate in a second. You'll see that I have quick demand for energy versus slow demand, which might not be always the answer that you get in other courses. We'll talk about that in a second. Okay. All right, so this is kind of the broad or the more detailed steps of the glycolytic system. Okay. And we can see the difference between using glycogen versus using glucose. And we can count up the ATP here. The big difference that we see there is that we actually use 1 ATP when we start with glycogen. If we start with glucose, we use 2 ATP. So actually, like that's ATP loss instead of gain, right? In both of them, we gain 2 here, gain 2 here. So we gain 4, right? But if we started with glucose, we only have a net of 2. We started with glycogen and a net of 3. It takes an ATP. And actually ATP when we start with glucose. Okay. No oxygen here. Total ATP made is 4, like I just said. Number used is either 1 or 2, depending on glucose or glycogen. Okay, and then as you just kind of answered on the quiz, we kind of talked about how long does this one last? Yeah, it's somewhere around 1 to 2 minutes, 30 to 40 seconds as well. It's an all-out sprint. The reason I like doing the 40-second wing gait is because of the sheer maximal lactate buildup, whereas the 30-second one doesn't necessarily always get you there. 40 seconds definitely does. And yeah. Okay, so what kind of activities would you say exemplify the use of this one? 2 minutes will be something, an activity that would last 1 to 2 minutes, like an all-out 1 to 2 minute activity. Okay. Sprint, yeah, what kind of sprint? Yeah, exactly. 400, 800, something like that. Even if I do it fast, but 2 minutes. But still, right around those middle distance type events are the ones that really exemplify the use of this. Yeah.
What are your analysis?
That's a good question. Yeah. Like a kind of floor routine. Yeah.
Or
How long do those typically last?
hopefully can be every
1 to 2 minutes. Yeah.
It could be longer.
Yeah, I think that's a great one. Yeah, track's always the easiest one because it's so linear. You know, this that's as long as that's the thing. But yeah, there's tons of other applications for sure. And I like that one.
Does each system have different recovery times?
Yes, for sure. So for example, the creatine phosphate system takes about 90 seconds to 2 minutes to replenish, which is a lot of what drives the science behind rest periods with resistance training. Right? It's about two minutes is what's recommended between sets for resistance training. Right? And a lot of that is based on the fact that it takes about that long for the creatine phosphate system. If you do something like a all-out 1 to 2 minute sprint, and you build up a bunch of lactate in your system, that can take a lot longer. That can take some, I mean, not too much longer, but somewhere around 5 minutes sometimes to get your pH back to normal. Really push it hard. So one of the things with sprint training is trying to make sure you don't do that too early on if you want to do a lot of the sprints, because if you do like, first sprint is a 2-minute sprint, then you've got a long period of time before you're really recovering from that because of that drop in pH happening in the cellular system. So yeah, so that kind of answers your question. Yeah. Other questions on that? Okay. What's the rate limiting enzyme on the glycolytic system? Remember? Okay, awesome. Got PFK or phosphofructokinase. Yeah, and so what the first concept map assignment in this class will be mapping out those 3 energy systems. And I definitely want you to include the rate limiting enzymes that we talked about here, as well as those key enzymes like PDH and LDH that I mentioned in the last slide, creatine kinase convention. And it's always interesting. So the idea of the rate limiting enzyme is kind of a weak link in the chain, right? I mean, it's the kind of slowest step, the one that we definitely need to speed up if we want to speed up this system, right? And so then the question is like, what stimulates this PFK to get going, right? And so very similar to what we just talked about, it's the same thing that I talked about for the phosphate system. If we start building up ATP and phosphate, that activates PFK and gets it moving, right? It activates a lot of the enzymes along the way, but that gets this whole process going when we have broken down ATP. Makes perfect sense because that means we need to make more ATP. So I'll move it. Okay. Any questions on that? And one thing, if you just to make sure we're on the same page here, at this point, after this point in the system, actually before that, this step before, it splits into 2, right? So we have 2 of everything after that point, and so that's why we end up down here with 2 pyruvates. We'll also end up with 2 lactic acids. If pyruvate goes through PDH, we have 2 acetyl-CoA, 2 turns into carb cycle. But it's all kind of at this point where we split from just getting 1 substrate into 2. So, okay. All right. Yeah. So talk about this question for a second with the people on your team. We'll come back in a second.
I read one, but I was thinking, what makes it work?
All right. What do you guys think? Go ahead and hold up your answer. Okay, we got some A, some B. Okay, we're a little bit split there. Okay, so when does delayed onset muscle soreness or DOMT? Like how long after the workout? 2 days. It's like 48 hours, even to 72 hours sometimes, so 2 to 3 days after the workout. Okay. What we know actually from measuring lactate levels in the blood is that it only takes about 2 hours to get lactate out of your system. Okay? You need your lactate level to get back to normal in 2 hours max, and that's when you've done something super intense. Most of the time, it's within a matter of minutes that your lactate level is back to normal. But for sure, many years, this is what we thought the DOMS was caused by lactate, right? And so many well-meaning coaches told you to go out and do a lactate run when you're sore, right? Go work all the lactate out a couple days after. And that was good. That probably helped you, but it wasn't because of lactate. It was because of the inflammation you had in your body and increased your circulation helped move some of that inflammation around and boost you up. But the lactate was gone a long time. And so not DOMS, but this one, yeah, the burning during and even seconds after an intense workout, that's going to definitely be caused by lactate, and really more the hydrogen ion that lactate releases, right? And so hours after, no, that's too long, but definitely a little bit of this stuff for sure, right? Because as we're working out and our pH is dropping because of release of lactic acid, those hydrogen ions will go and bind to all of the enzymes right here, right? So they go back, they start building up those hydrogen ions, they start binding to all of these enzymes, and they start making it so they don't work as well, right? And this is what I was talking about with like a negative feedback system, with the glycolytic system. The more lactate we make, the harder it is to make lactate, right? Because it slows down the system, which is actually perfect for us, right? Because we don't want to drive our pH into too low because we can really damage ourselves. And secondly, this is a total waste of energy too, right? We only get 2 ATP, whereas we could be getting 32 instead, right? So the body really doesn't want to do this if it doesn't have to. And so it's got this built-in feedback mechanisms to, yeah, we can do this a little bit, but I'm not going to do this for very long because you're wasting energy and you hurt yourself. Right? And so that's really what this is. So those hydrogen ions go back and they bind to all of these, kind of start to cause a little bit of deviation, making it so this glycolytic system slows down drastically. So that would feel like weakness. That's what that would feel like for us. If we can't make as much ATP as we know, that would feel like kind of weak, where we don't have much energy, right? So both of these would be. Questions on that? Okay, so let's look at this idea of this fork in the road for pyruvate, right? We talked about how pyruvate's got to go either through LDH, which turns it into lactic acid, or PDH, which turns it into acetyl-CoA, and that's going to dictate which system we use. And so the way I like to think about this is in terms of the funnel. Okay, so if we think of PDH, that pyruvate dehydrogenase, as a funnel, then this is kind of a good analogy for this. Does anybody use a funnel? Like maybe you change your own oil in your car, or you maybe funnel cooking in some way. Somebody has some experience with a funnel, so you know that the width of this part of the funnel is really kind of dictating the maximal flow that funnel can handle, right? And so you start pouring something into a funnel, and if you just take something and take a gallon of water and just dump it all into the funnel at the same time, it's going to overflow, right? There's still going to be some coming out the bottom, but you're putting in more fluid than that funnel can handle. You're kind of exceeding the maximal rate of that funnel. But if you start pouring it slowly, you see the fluid starts building up, then you got to kind of slow down. It's still coming out the bottom that whole time, but we can keep putting it in, right? And so it's kind of the same idea with this pyruvate dehydrogenase enzyme, is that, and so we'll do it in terms of kind of like, let's just say we're going to go from standing to all of a sudden walking, okay? So when we do that, we got all that stored ATP in our muscle, right? We're going to start contracting muscle, we're going to start activating our myosin ATPase, so that breaks down some of that stored ATP, right? That results in some free phosphate, free ATP program that activates our glycolytic system. So all of a sudden we start getting a bunch of pyruvate starting to come down because we just used our glycolytic system to do that, or glycolysis to do that. Okay, so pyruvate levels build up a little bit, right? So this is like you starting to pour something into the funnel, but you're doing it in kind of a controlled fashion. So we're getting some pyruvate hitting pyruvate dehydrogenase. Okay, we're not getting too much, but we're getting some. Okay, so most of the pyruvate that we're building up can be handled by the funnel size of pyruvate dehydrogenase. Okay, there's still going to be a little bit of lactate production. Some of it's still going to go through LDH, but for the most part, most of it can go this direction, go to acetyl-CoA, go to the first. Okay, that's for the walk, right? That was when we just start walking, okay? But then we all of a sudden go from standing to instead we just start sprinting as hard as we can, right? That takes a lot more energy, that takes a lot more ATP inside of our muscle cells to do that. So all of a sudden we just created a ton of ATP and phosphate, which means we really activate all of those enzymes in the glycolytic system. So instead of just a little bit of pyruvate, we've got a ton of pyruvate coming down the pipe now, right? Because we just activated that glycolytic system, broke down a bunch of glycogen. Here comes a bunch of pyruvate. Okay, this is you just dumping the bucket, you're dumping the gallon jug into the funnel, right? There's no way that the pyruvate dehydrogenase we have can keep up with that. So what happens is a lot of it overflows and goes this direction. Right, we've got to start using LPH to do that. Sure, still some of it's going to be coming this direction. We're still using our oxidative system, but just not very much in comparison. And so the vast majority of it has to go this direction, kind of an overflow route to make us energy quickly. Okay, and so that's really kind of the bottom line with this. What's going to dictate which way pyruvate goes is how fast we need that energy, and how forceful of contraction our materials. Okay. Now, for sure, when you in the biology textbooks, it's always gonna say it's about the presence of oxygen in the mouth, which is absolutely true. If you don't have oxygen, your cells can't do the oxidative system. But there's lots of lines of evidence to show us that there's always oxygen in our cells. We're not like a single cell in a petri dish. We're a multicellular organism. Our brain's never going to let us deplete ourselves with oxygen completely. We make lactate at rest. We make lactate all the time. There's lots of different lines of evidence to show us that it's not really about oxygen or not. It's about how fast we move the energy. So that's kind of what's going to drive this direction that pyruvate goes. Okay. Questions about that? All right. Well, we'll stop there and we'll continue this stuff on Friday. Good to be with you guys. Have a good rest of the day.